Okay, indeed. And so it's my pleasure to spend the next few minutes to discuss really novel paradigms in colon. And it's, I think, important that everybody gets familiar with them because the point is to change our therapeutic approaches to develop drugs to address these novel paradigms and to incorporate them in our clinic. So it's really a community effort. And so there's a paradigm, I think, that at least my generation was brought up on, which we can refer to as the Vogelgram, but it really assumes a very linear kind of progression between adenoma to carcinoma with the addition of certain oncogenes driving that progression. And those oncogenes and that paradigm have really been the target of our therapeutic approaches in the last few years. So anything that was progressing you along the line should be shut down. But at this point, we rather think the model is a bit different, as should then the drug approaches be. I was just going to wiggle with my screen here a little bit. But so I'm going to give you some really bizarre biological lessons, but this one overlaps with the previous speaker, which is nice. But they're fundamental lessons to understand the novel approach to colon cancer. So this part is that you are indeed formed, as the previous speaker showed, as foregut, midgut, hindgut in the beginning. And what is important are these two messages that I highlighted on the left. One, there's a very specific code for these regions. So each of these regions has what I would call the Hox code. And the Hox code really determines certain cell states and plasticity in the colon. So you not necessarily have the same cell types or ability to be certain cell types, more an ability to be certain cell types in the different parts of the colon. So that's what I'm talking about as the Hox code. And it's going to be important. The other thing I want to highlight on this slide is the CDX2. The CDX2 belongs to the midgut and the hindgut. It's really what determines this midgut, hindgut away from the foregut. You know that your pathologists use it a lot to say when you have a tumor of unidentified origin, they do the CDX2 staining and they go, oh, it's a colon tumor instead of a stomach tumor. So very important that CDX2 is something which really aligns with hindgut and midgut. And I'll show you examples of where that kind of clashes with our current paradigm. So the Hox code allowing a developmental plasticity of cells, and then CDX2 as a marker of hindgut and midgut. And the current model, again, really very much, I just have to see where I can put this away. Move it to the top, there we go. So we have these regular little crypts in our colon, and I think we all are familiar with the concept that there's a stem cell, that feeds each of these little crypts, with the upper cells just dying, doing nothing, dying after a few days, but a continuous replenishment by that very famous LGR5 stem cell shown here in green. And that LGR5 stem cell is the only one that really acts like a stem cell population here, more or less, but it is actually, its identity is determined here by its position and by the feedback it's getting here from these specialized fibroblasts. So the stem cell is a state we find here, and it is dependent in its natural state on the signals it's getting, the Wnt signal it's getting from these fibroblasts. So that's very important. It's something which is situational, highly regulated a zillion times in our colon, and means that our colon doesn't form polyps, etc. Just regulated stem cells in a position, upper cells die, and that's a very nice system. Again, the LGR5 stem cell giving off all the daughter cells which terminally differentiate and have a limited lifespan. Do note that there's this kind of mysterious little putative other stem cell, which I'll come back to later, and which is really the new paradigm. Okay, but so this is what we classically know. And in comes this first oncogene-related topic is this APC mutation, because we all know colon cancer always has APC mutations. Familial colon cancer is driven by a germline APC mutation. But so what is this APC mutation? Why does it disrupt the system? It's simply because APC mutation equals Wnt activation. So now, instead of being dependent on your fibroblast for your Wnt activation and your stem-like behavior, you can do this anywhere. You just get an APC mutation here, it's fine. You will be an LGR5 cell. You will create a novel little crypt. Okay, so these APC mutations, which are induced in our colon, amongst other by some E. coli strains, they actually create fake little LGR5 situations along positions where they should not be, these cells. And so very plastically, this can look like that. You had your normal crypt, but you have an APC mutation here. And it's just going to kind of form a next little LGR5 compartment and create cells on its own, and we can start to see that even endoscopically. And these cells can even expand and take over the next scripts, and in the end, we sit there with this beautiful little APC-driven polyp. Yeah. And by definition, all these cells should be LGR5 because through this APC mutation, they have just gained the autonomy to be LGR5 anywhere they want. And that's also what we see. So I don't know if the red will show very well on the screen, but here's the little LGR5 dots, which should be in the bottom of your normal crypts. But as we have a polyp here, all cells are LGR5 positive, and it's not restricted to the bottom of the crypt. And so that is typically one way of becoming a tumor. And it's really hijacking a stem cell compartment, which has to exist in every crypt, but then amplifying it multiple times through mutation, creating a fake crypt all over again. But that is one way, okay? And there's other ways. That this is again showing the one way it can become quite massive. This you see is a larger polyp. It's tubulovillous adenoma. Each of these cells, the yellow is LGR5, is packed with LGR5, knowing that this should be a phenotype of just 2-3 cells at the bottom of the crypt. So quite a powerful way to develop things. And when we have to think about the immune responses to this and how we're going to target all that, you have to really get into that mode of this is a Wind LGR5 driven disease. It's recapitulating the bottom of the crypt. And that's also how we're going to have to drug it, both from the immunological part as from whatever cell dying component we want to give to it. And quite different than the rest, as I will show you. So again, a visual on this same kind of LGR5 tubular adenoma Wind-driven pathway. So this would be tubular adenoma. It would have an APC mutation. And I'm going to show you this because I'm going to use these colors on the next slide. So you would expect everybody looks pretty tubular, like little crypts on steroids. You have this CDX2, which is the hindgut and the midgut, so colon. It's nicely there. Every nucleus has a beautiful little CDX2 signal, purple. You see the yellow are your goblet cells with the MUC2 beautifully there. And notice the absence of the green color on this slide, green being MUC5, which is a non-colon mucin. So you wouldn't have expect to see this in this colon lesion. MUC5 is a gastric mucin and you don't see it. Okay, so I think up to here, I hope I've convinced you that that kind of classical Wind APC LGR5 dominant phenotype does exist. I would argue it's a minority of what we deal with in colon cancer. So what else exists? And the visual here, I think, is quite striking. Here's a tubular adenoma we spoke about, but here's another type of polyp. It's called a serrated polyp. And you notice the difference is quite striking. The normal colon here has the CDX2 purple, has the yellow goblet cells, MUC2, but look at the lesion itself. The lesion has no CDX2, so it has no feature of colon anymore. It has also gained this green mucin, which is a foregut mucin. So it's lost the hindgut markers, it's gained the foregut markers. The cells, I don't think you can see it from distance, don't grow tubular at all. They grow serrated, they do lateral invaginations. It's a completely different beast and way of growing. It also doesn't grow up like a polyp, it just grows flat over your colon, and it actually has horizontal tension in the cell instead of upward tension. So completely different thing. I'm not going to bore you because it's beautifully published by Ken Lau, but if you look at it by single cell, up here is a tubular adenoma. I highlighted it here. These genes are off, these genes are on, and the genes that are on in tubular adenoma are all your regular ones, your little LGR5, your little Wind, your everything that is to do with your regular colon Wind activation. But you can appreciate that in the sessile serrated tumor lesion, it's quite the reverse. None of these genes are present, and a whole set of different genes that I'll discuss are on. So we have something that has actually lost a colon lineage. And the clinicians in the audience, maybe we are very used to Barrett's esophagus, where we go like, oh, Barrett, it's an intestinal metaplasia in my esophagus. Well, this is actually gastric metaplasia in your colon. And it's we are going to call it gastric, but also fetal reversion. It's back to a, I would say, ancestral state, and that's important. So then they don't care about this mechanism. They don't have APC mutations. They don't have LGR5. They don't have CDX2. They have nothing of this going on. So where do they come from? Do they come from these other little cells that we always thought were there? Yes, they do. So where do they come from? They exist, they're green, yeah, they have this non-CDX2 state, so they're kind of certainly hovering somewhere else than over here, yeah. And that Hox code, as I showed you, it's too detailed to explain now, but it's going to be important and quite different in these cells compared to the others. And so again, giving some more visual examples, it's easier to understand like that. So here's the classical Wind-driven tumor, because I showed you polyps, but what about the tumors? here's this APC mutant, classical, CDX2 positive. You see the purple everywhere, nicely tubular adenoma kind of shaped. It's a tumor, but it has this still a bit of a glandular kind of way of growing. And so that is, again, your quite rare APC wind-driven, CDX2 positive, quite prototypical colon tumor, but actually rare. Yeah, we call it I2. And this one, which under the microscope, you see looks very similar when you don't do all this coloring and you just look by H&E. This one you see, you can appreciate, has lost CDX2. There is a staining that was applied on this slide, and you see it doesn't pick up the cells, so they don't have the hindgut markers anymore. They've gained the green mucins. Also, their goblet cells, you see, can form very bizarre little immature bubbles of mucin. So this is actually a adenocarcinoma of your colon, but where the majority of the cells are actually in a fetal foregut state, something we do not differentiate by H&E analysis, morphological. And many tumors arise to a mixed state. So even if they departed in this one, they will end up still getting mixed up like this. So where, again, you have your normal colon, your CDX2 in your normal colon, and here's your tumor with purple areas which have a colon lineage and green non-purple areas which have the more fetal lineage. They intermix. Here's a nice little front of T cells colored. So you have these really organized mixes, and you say they don't look organized, maybe a little bit, but not much. But don't forget, that's also a problem of our current histology. It's not only that we don't do these stains, so we can't see that these cells are heterogeneous, but also that we have always a 2D plane, but these things are organized. In maybe another speech, I could show you all the spatial transcriptomics on this, but these things are really organized structures growing in layers. And so there's a very strong mechanobiological determinants of cell states, and those are underappreciated when you just have this kind of 2D state. But so the mixed ones, the majority of tumors are actually like this, and that has serious implications for therapy. Yeah. So I wanted to have a small summary of the previous points before I move on. So I kind of have told you that there are at least 2 of these epithelial cell states in colon. Roughly, one could say the colonic CDX2 positive version, and then roughly one could say the fetal gastric in orange, the other version. Okay, this work is published so that you can look that up if you want more details. But what is also important, so we got our colonic and we got our more fetal gastric one. But what's important is that the star of colon, the MSI high tumors, they are in the fetal group. Okay, they are here. So that's very important now to look at their sister, who is also in the fetal group, but is not MSI high. It's MSS. So we have a whole set of colorectal tumors, whether they're MSI or MSS, which actually share these fetal features. And that's very, gives us a lot of hope on how we're going to immunologically target these, which already share this kind of fetal code with the MSI. Yeah, we have these purely non-fetal. They're quite rare, and I would say that the mixed tumors that I showed you, they're the majority. So they would be sitting kind of here in the middle. Yeah, so pure MSI, pure fetal, mixed fetal, the majority, I would say, of colon cancer, and then these are rare. So our upcoming work that we've submitted will really show, well, these distributions and further definitions of these cells. Also, they are not completely strange to us because already MSI, we know, is always here. I think this split within MSS is a relevant one and it's a new one, but also some mutations we often use, such as BRAF. BRAFs will always be here, always in this group, like the NRASs, the very strange KRASs, the codon 46, etc. They will all be here. Everybody here will have a KRAS or a BRAF, yeah? So you're always really playing with that MAP kinase angle on these fetal cells. Whereas here you can live with just an APC, for example, and traditional copy number changes. So even our genotypes are quite useful actually already in departitioning these things, but not perfect. I don't have 100% time to do that now, but it's not perfect, but there is a relationship. So also, when are these scary cells appearing? Because they seem quite different, and how do we deal with them? And so just going to, why did nature actually maybe even keep these cells? How do they pop up all of a sudden? And one of the reasons is because it's normal injury repair requires them. So this is a really cool review by Ophir Klein. I just want to highlight again what you know. On the left is your normal crypt. Notice your LGR5 cell sitting there. So this wind-driven, positionally, everything's perfect. LGR5 cell giving off the daughter cells. Notice here that you have then an insult. I'll come back to that. And that insult really breaks down a whole structure. It not only removes your mechanobiological cues, you see your basal membrane, your fibril rest, a huge ECM remodeling, which is very dangerous, as I showed you, and these kind of areas are actually determined very much by that, but it also brings in a lot of inflammatory cells and inflammatory cytokines, and that is actually killing off these LGR5 cells. It's too hot for them to stand. You see they're gone, the green ones, but so you have to deal with this, and that's when pop up these kind of reserve fetal cells, these fetal cells during normal injury. Yeah, they are determined by completely different pathways. These cells are exactly in normal injury, are exactly the cells we see in the polyps and the tumors. So instead of stopping the mechanism and getting everything reorganized, this step is not happening, and these are exactly the cells that we're getting. And of course now mutations are also able to produce these cells. Yeah, but so interesting that the ability to either work with this system or with this system is actually ingrained, and it is certainly triggered and very much a feature of homeostasis in these very hot and difficult environments where all cues are lost. These are the cells that are able to act without these cues. And there's a whole set of mechanobiological lessons in these cells, which we hope to exploit in cancer treatment, because if they can arise, we can probably make them de-arise also. Yeah. I was giving a speech yesterday to radiotherapists, and they were pretty scared that irradiation can actually cause some of these lesions. And I think that's also an important thing. Are we sometimes causing some of these cycles where we're making these cells more potent or appear? Something to think about, and we need a lot more talks to go through that. Also, another very nice review here, very recent, but which summarizes again that these fetal cells are really now there to stay, and they are seen in many different situations. And I'm not going to go in detail. I already said that during infections, etc., you see them pop up. You can also get them there by doing manipulations of your extracellular matrix. So as I said, this kind of mechanobiological determination of these cells is very strong, and you can change your collagens and everything, and you can have them pop up, yeah, which is really interesting. This is to say that they're seen a lot in tumors, and that in organoids, recent work, you can really also make them appear by changing the growth conditions, for example. So it's an inborn kind of state that is there, and that can appear in certain conditions, and does. And this summarizes a little bit of what this means for us clinically. So they are certainly there already in the earliest polyp lesions. These are tumors, these are metastases. In the tumors, this is also a paper we submitted now, but in tumors, we really see that there's two axes, basically. There's the wind type of behavior, and there's the feet, so the wind here, and there's the yap or fetal type of behavior. And all tumors and all patients contain mostly a mixture of these cells, with some exceptions, such as like BRAF would be mostly YAP, and an APC only will be mostly wind. But otherwise, we're really dealing with a sparkly star of adaptive cells, and that is also why our therapies have been so difficult, but also sometimes maybe therapies, I don't know, could have some negative effects at some points. What Karuna Ganesh recently published in Nature, I think a week or two ago, and this is at the old bioarchive link still, but is that as you go to metastasis, and she compares primary and metastasis, you're actually really pushing that fetal angle. So by the time you're in your metastasis, you are really much more like a fetal endodermal developmental cell than anything else. So certainly when we're in metastasis, we're going to have to learn to deal with how to kill those fetal cells. Yeah. Whereas in tumors and polyps, we can still get away with dealing with these other two populations. And they each have different treatments. So that's a bit scary. Also, they're very early. They appear easily in these lesions. This is a resected stage 2 or 3 colon cancer. So they're really there quite early in these mixed states. And certainly what can trigger them is injury repair cycles. I'm not going to show you the data on selective chemo killing, but I can just hint that, if your purple cell is more sensitive to chemo and you eradicate it, well, you're just making space for your green one. It sounds very logical. And also your stromal supportive structure may all of a sudden become very positive towards these more fetal cells. Radiotherapy also, I've showed you some hints. And I'll just show you two details of how they rely on stromal features, some already quite well known, and also how this impacts your immune system. So I'm going to move on to those two small topics, but I'm just going to gloss over them. But the point here that I want to say is that they rely also and are co-determined by these stromal and mechanobiological features. And the beautiful article that you should read, and we have no time to go over it now, very recent, from the group of Jorge Moscat, he will show you that these cells, they creep down the structures once you create them. He creates them through APTC, but that is not so relevant for my message now, that they are really having this behavior of coming like to heal a wound, and that they are very dependent on where they are. And his paper the year before showed that you can actually kill these cells by blocking their hyaluronic acid scaffold, which they so much enjoy during their positional changes. So beautiful work in mouse, really proving now some of these concepts that those cells exist and that they are elicited by these wound-like situations, but also giving us a tool on how to eliminate them because they are dependent on their structures. Also work that you may know from 2 years ago from Eduardo Batley, where he made a mouse, again, with an APC and a KRAS mutation. So it had both, let's say, capacities. And you could see there that the Wind cells, as LGR5 cells here, the green LGR5 Wind, they're sitting more in the tumor core. As you go a bit towards the invasive front, you tend to have more of these red cells, still some green. And as you're reading those tumor buds, you get these red cells. And so these are the reds, are the fetal, they're called EMP1 here, but these are these fetal ones. And then these are the LGR5 Wind ones. So also it's important that they arise during and are the cause of a lot of the metastasis, of the majority of the metastasis. So you can track them, these red cells, they're actually leaving the tumor and enter the circulation. And what was also important is that when they arrive in the liver here, you have a little red cell now that has arrived in the liver, they magically, and will have to actually to grow, retransform into their other phenotype. So you have these kind of shape-shifting cells. Also, this is one genotype of cells, but they shape-shift, okay, green, red, and then red to green again to be able to grow a mass, which is otherwise just a micromet versus a macromet, yeah? And they organize themselves quite well in that. So the thing is here that even these mutations, that's why I hinted that mutations are not perfect, a mutation is necessary for, but is not enough, or there's not a one link between phenotype and mutations, but certain mutations are necessary for the shape-shifting ability, and certainly the KRASs are important, but that's so they can override an APC signal at this point and then let the APC signal be dominant again here. So all kinds of completely different than what we thought, and the implications of that, again, beautiful here, this paper where Tyler Jacks asked, okay, a hallmark of cancer is to avoid the immune destruction. How can I model that? So takes, again, an AK, it's no coincidence, again, a mouse that has the A and the K, and grows a regular organoid out of the colon of such a mouse, outside of the mouse. Here, the oncogenes are not yet activated. Here, they're activated, so it doesn't matter. Now we have just this little AK organoid growing outside of the mouse. So it doesn't see an immune system. It's just enjoying the full glory of being A and K and P53. As you inject it orthotopically in the colon of the mouse, of an immune-competent mouse, it will see an immune system for the first time. And it's very interesting to wonder how this AK clone is going to react to the immune system. So either it'll die or whatever grows and is visible and you can harvest has now adapted to an immune system. So you can really compare the AK adaptation to the immune system. And when you do that, without going into detail, just you have this volcano plot, you see that these guys have gone completely fetal. Trust me, this is the fetal. Remember the clustering we even saw it once. So this is fetal. So again, this ability of this APC KRAS, where initially they were more Wind phenotyped and to go fetal is quite strikingly proven, and it's what we see over and over in the tumors. And in this case, a little side story, which I think is important, is that while doing that fetal reversion here, one of the fetal genes is SOC17, so which is normally off in your normal colon, on in the fetal colon, but so now got upregulated again in this fetal transversion. And SOC17 in the paper, they show beautifully, just focused on the bottom, it actually stops the interferon gamma receptor. So it actually stops the transcription, it inhibits that receptor, meaning that you have your epithelial cell now, which is fetal, and also is quite insensitive now to T cells. You have you don't perceive, and you have much lower levels of interferon gamma. Your MHC is down, and you can see that your T cell is just basically going to be very exhausted looking at at this epithelial fetal cell. And what is interesting, the mouse, don't forget, had an APC and a KRAS mutation, so it had both fetal and non-fetal cells. And you can see that there's quite an organization going on with the fetal cells and the low interferon on the outside, and the more wind-driven LGR5 ones kind of huddling in the middle as a protection against the T cells. So quite sneaky organizations over and over, and a quite sneaky plasticity, which is functional and really is aiming at the survival. Okay, so that really means that we're going to have to deal with that as we look at our immunotherapies. And immunotherapies come in multiple shapes and forms. The more classical are things that are still CD8, let's say, focused. And so when we did that and just took colon cancer and took all the CD8 cells out and just did a CD8 focused view of colon cancer, and this one is published also if you want more details, then it became obvious, a few things became obvious also from the CD8 view. So both from the epithelial view, we would have said that there's not one type of MSS colon cancer, nor would we say that from a CD8 view. Yeah. So in the MSS, and it's not unexpected, we see CD8 cells that look like these states, and we see CD8 cells that look like those states. And these are much more associated to these YAP inflammatory type fetal cells states. And these much more to the APC, LGR5 type states. And there's a lot of mixes going on. So the work that really has to be done now is to get all the epithelial and the CD8s all matched in the mixed, which is predominant. Extremes are fun for research, but most patients are not extreme. By the way, and this is also clear in MSI, there are two epithelia. This is work that will be published soon, but there's also two types of CD8 states. And there's a lot of other cells that matter. So this was, the more classical type of cell, but there's a lot of other cells that react to these epithelial states. But so take-home message, you can see it even as you look at the immune cells. And so for me, where we can focus now for the development of immunotherapy is really thinking of which are those immune cells that interact, drive, and then deprogram also these fetal states. This is going to be the key, just looking from the normal situation of where these cells arise and how they arise and how they disappear. And so I think there's a package of novel therapies directly targeting immune cells and extracellular matrix components that we're going to be able to develop. And of course, this is normal wound healing. And I would really develop my drugs according to this axis. But of course, we have then some mutations that have accumulated in the polyps and the tumors, but they're pretty easy to target because we got our MEK inhibitors, we got our BRAF inhibitors, and of course, they don't do anything on their own, as we are now learning, but they will be extremely helpful in these combinations. So combo, but very rational combos, quite different than the ones we're doing now. Yeah, and I want to end up slowly. Because the thing is I just want to pitch neoadjuvant, because everything I told you is if you want to have your immune system attack these cells and have some memory while doing that, it's much better to treat this before surgery when all this mess is there. So while the tumor in its full glory is there, and you would give the correct immune therapies. I just talked about what I think are correct immune therapies. And you could then grow a very nice memory situation around that. You can then still resect your tumor, but at least you'll have the benefits of that memory. So in addition, I think, to developing the right drugs that really attack these epithelial and immune states specifically, we will need also to change the moment that we are using them. Okay, and so I think if we really want to avoid meds, that neoadjuvant approach with creating long-term memories is going to be very important. So that's I think our responsibility. And I'm going to finish up by saying that, of course, as they become metastatic, and if we allow them to become metastatic, we're dealing with quite a difficult situation. You just saw that they go from local fetal now to fetal in the liver. And I'm just highlighting again what Karuna had showed, that you really move to these kind of fetal states in the liver. And that is even more difficult to target, because the fetal states of the colon, they have a logic to it, and we can understand that one. The fetal states in the liver have undergone probably more transitions, and there's all kinds of interesting immune exclusion states. Look at the poor CD8 cells hanging around, they can't get into the metastasis. And so those will also take a lot of work. And at the moment, I would try to avoid these states. But for sure, the way we're going to approach them will have to be different than the way we approach the primary tumors. And I just want to thank all my teams for this work. So thank you very much.